Study time with Nabilah.
Hello students, so today our topic is substances across plasma membrane. So today our main focus is to learn about the structure and function of plasma membrane, comparison of passive and active transport, and also the types of passive transport. Did you guys know what is plasma membrane, the structure or the function of plasma membrane? So, plasma membrane, also called as cell membrane, is the membrane found in all cells that separates the interior of the cell from the outside environment. So, does it have in the plants or bacteria? Yes, it is. Because in bacterial and plant cells, a cell wall is attached to the plasma membrane on its outside surface. So, the plasma membrane consists of a lipid bilayer that is semi-permeable. So, did you know that this unique structure of plasma membrane is known as Rafid mosaic model? The plasma membrane is composed of phospholipid bilayer, which is two layers of phospholipid back to back. Phospholipids are lipids with a phosphate group attached to them. The phospholipids have one head and two tails. The head is polar and hydrophobic or water-loving, but the tails are nonpolar and hydrophobic or water-fearing. So, the head will face outside while the tail will face the inside. So, these phospholipids will line up in two layers with the head pointing outward and the tails hidden in the middle. So it's basically look like a sandwich. So now let's move on to other structure of plasma membrane. Okay, we'll look at the carbohydrate, glycolipids, and also glycoprotein. So all of these are carbohydrate, but for glycolipid and also glycoprotein, it is a combination between carbohydrate. For glycoprotein, it is combination between carbohydrate and also protein, while the glycolipid is the combination between a carbohydrate and also lipid. So, what are their functions? They are functioning in the cell recognitions for other foreign substances to enter the plasma membrane. Next, the function of cholesterol on the plasma membrane is to stabilize or immobilize the structure of plasma membrane itself. The integral protein functioning as the transport molecules which allow substances through it. So, peripheral protein act as the identity marker at the cell surface which for antigens. And now we'll look at the channel protein, which also works the same ways as integral protein, which allow the molecule to transport or to go through it. It helps those substances to enter the plasma membranes. Alpha helix protein is one of the integral protein which works the same way, which allows certain particular molecules to enter it or to enter through the plasma membrane. And lastly, the globular protein, it functioning in the metabolic functions. Okay students, so now let us do a simple activity. Can you take out a piece of A4 paper and also a pencil? Let's draw and label the plasma membrane together. So, here are the answers for the previous activity. Did you get all correct? Well done! What are actually the main function of plasma membrane? So here, there are 4 functions of plasma membrane. First, it is selective permeability. Since plasma membrane is semi-permeable, which only allows certain substances, so it helps in transport those substances across the membrane. Second, it is for digestive. Plasma membrane takes food in and excrete waste product out of the cell. And third, it is for protection. Plasma membrane forms as the outermost boundary of the cells. It protects the cells from the foreign substances to enter it. And fourth, it acts as cell recognition. The plasma membrane contains cell surface receptors. As an example, the glycoprotein receptor, which presents on the red blood cells. Next, we will take a look at a simple example of the experiment on this plasma membrane, the whisking tube experiment. So, let's take a look.
Alex and his friends are having snacks. Alex is eating rather fast without chewing his food properly. His friends suggest that he chew his food slowly so that it will break down into smaller pieces. Some food molecules are large, such as starch, while others are small, such as glucose. This can be shown in an experiment using whisking tubes. One whisking tube contains a starch solution, while the other contains a glucose solution. Both whisking tubes are placed in boiling tubes with distilled water. The whisking tubes are left for 30 minutes and then tested for starch, iodine test, and glucose, Benedict's test. It can be shown that when iodine solution is added to a sample in test tube P, it remains brown in color. This shows that starch is absent in the distilled water. The starch molecules are not able to pass through the whisking tube. Benedict's test is used for a sample in test tube Q. The Benedict solution changes from blue to a brick red precipitate upon heating. This shows that glucose is present in the distilled water. Glucose molecules are able to pass through the whisking tube. The whisking tube in the experiment acts like the small intestine in our body. The digested food, like glucose, are absorbed in the small intestine and diffused into the blood, which is represented by the distilled water.
There are 2 types of transportation of substances across plasma membrane. There are active transport and also passive transport. So here is the differentiation table between the passive transport and also the active transport. Let us take a look for the definition of passive transport and active transport first. For the passive transport, it allows substances move down its concentration gradient from high concentration to low concentration. Meanwhile, active transport allows substances move against its concentration gradient from low to high concentration. Passive transport does not require cellular energy to undergo the transportation. Meanwhile, the active transport requires cellular energy in the form of ATP to undergo its transportation. So, the substances that can move with the passive transport are the water-soluble molecules like small monosaccharide, lipids, carbon dioxide, oxygen, and water. Meanwhile, active transport, ions, large protein, complex sugars, and cells are transported through it. And there are 3 types of passive transport. which are simple diffusion, facilitated diffusion, and also osmosis. And for active transport, there are only two, which are sodium potassium pump and also bulk transport.
Now let us do simple second activity. Think and click. There are three multiple choice questions. Choose the best and correct answer for each question. First, blanks transport use ATP for cellular energy. A. Active. B. Passive. Second, which of the following types of transport belongs to passive transport? A. Osmosis, bulk transport, and simple diffusion. B. Facilitated diffusion, osmosis, and simple diffusion. C. Facilitated diffusion, simple diffusion, and sodium potassium pump. Third question. Substances move against its concentration gradient from low to high concentration is the definition of the blank space. A. Passive transport. B. Active transport.
So here we will take a look at the passive transport. There are only three types of passive transport, which are simple diffusion, facilitated diffusion, and also osmosis. So first, let us take a look at the simple diffusion. So simple diffusion is the movement of small or unpolar molecules through membrane from high concentration to low concentration. As you can see here, there is no help from the carrier or no ATP is needed to undergo these simple diffusions. So the molecules which have high concentrated will move to the low concentrated region through the simple diffusions. So now facilitated diffusion. It is the movement of large or polar molecules through the protein channels from high concentration to low concentration. So it needs a facilitator or carrier to move the substances across the plasma membrane. There are two types of protein channel, which are carrier proteins, which transport molecules, and second, the channel proteins, which transport the ions. So for the last part of passive transport, which is osmosis. Osmosis is the movement of water molecules through the membrane from higher water potential region to lower water potential region. So osmosis usually refers to water, and the terms of water potential only can be used in the osmosis process. There are three types of solutions in osmosis, which are hypotonic solution, hypertonic solution, and isotonic solution. So here are the differentiation between the solutions in osmosis. First, the hypertonic solution. It is a solution that causes a cell to shrink because of osmosis. Second, the isotonic solutions. A solution that produces no change in cell volume because of osmosis. In a simple way, we can say that isotonic solution is in an equilibrium state. And for the third one is the hypotonic solution. It is a solution that causes a cell to swell because of osmosis. So this solution will cause different types for the cells in the animal and also the plant. So first we will take a look in the animal cells. For the hypertonic, the red blood cells will shrivel since the H2O or the water will acid the red blood cells. So it makes the red blood cells to shrivel. Like the isotonic, well, it is in an equilibrium state, so it is normal So next, the hypotonic, it will lyse or it will burst since there are more water molecules enter the red blood cells. So we will take a look at the plant cells. So in hypertonic solution, the plant cells will plasmolyze since the H2O or the water will acid the cells due to the more solute, which is high concentration of solute outside of the cells, then it makes the water molecules inside the cells to exit the cell. So it will plasmolyze. So isotonic, it will flexit, which is normal for the plant cell, but we don't call it normal, we call it as flexit. And for the hypotonic, the cell will become turgid since the solutes in the cells is higher than outside of the cells. So the water molecules will enter the cells and it will make the cell to become turgid.
Now, let us do activity 3. Create mind map for the passive transport.
Thank you for watching my videos. I hope that you can understand better for this topic. Don't forget to like, subscribe, turn on the notification and share to other people. And don't forget to watch my next videos. Hope that you have fun in learning biology and bye!